Hello viewers, welcome to my genetics lab project. You'll notice that this video has a little bit of a podcast feel to it as I have my friend Shauna here, a business major who has no idea what this project is about. So when I explain something, she'll ask questions that you, as the general audience, may also have. The slides have a lot of information on them that I'm going to do my best to summarize. So this semester, we were not able to attend class in person, so we did all of our labs from home. For some classes, this includes watching videos and filling in worksheets, but for this class, we were doing what everyone else seemed to be doing at the beginning of the pandemic, making sourdough. So we got some fruit, water, and flour and put them in cups and recorded daily observations, feeding them on a 24 or 12-hour schedule.
Wait, don't you normally put yeast in bread batter? And how do you feed bread?
Well, yes, but sourdough starters are different. One of the main points of this experiment was to use the natural yeast from the fruit and even the flour to allow them to arise. And feeding really means taking some of the starter out and mixing in new water and flour. Here's my experiment specifically. I had a cup with banana and another with grape, and the third was the control.
Did you just drop a whole grape into the cup?
I crushed the grape up, but left the skin in there. I did not put the peel of the banana in the banana cup, however. This is some straightforward information about the history of bananas and grapes. Grapes are mostly found in the form of adult beverages, while bananas are a nutritious snack that I personally enjoy every day. Kind of cool how the oldest grapevine is in the country is in our home state of North Carolina. The next slide focuses on people who have used these fruits: Babylonians, Hittites, and Indians, and how they make bread. The most similar style to ours was the Hittites because they did not use yeast either. Bread was much more important to them than it was to the Babylonians and the people of India, who mostly used it as a complementary side dish. I want to talk very briefly about my own experiment. My observations are on each slide if you want to pause and read through them. Week 1 was not very exciting, but week 2 got a bit more interesting when the starters became much more aromatic. The grape one was my favorite because it didn't smell disgusting like the other two, but rather like wine.
Not that you know what wines would smell like.
Oh, of course not. Week 3 was where it all ended. I have my short anecdote on this slide explaining how I lost most of my time with my 19 credits of classes and simply could not keep up with my starters, especially because I was traveling home a bit. Here's a main question of interest.
Good, because I have been wondering what all of this has to do with genetics.
Honestly, me too. But what we are interested in is the bacterial and fungal composition of the starters, which we found out using DNA sequencing. Keeping this as simple as possible, we are sequencing the DNA of the microorganisms which call our sourdough starters home. We used next-generation sequencing where specific regions of bacterial and fungal genes are sequenced.
Okay, that all makes sense. You're just determining which types of bacteria and fungi are in your starters. Kind of crazy if you think about it.
I know, this planet itself is so diverse, but the trillions of microbiomes around us are just as diverse. Maybe even more. Next was the rising experiment, but I had no starters to perform this experiment on, so I used the data from a friend of a friend. By the way, all sources of information can be found in the description of this video. These graphs show how much her starters grew over the course of a day.
Wow, they grew that much? Did she do anything differently?
Yeah, they were being fed more regularly for this experiment, but the next experiment is the big ticket. These were my 3 questions before sequencing. I wanted to know the similarities between berry starters, any patterns that could be deduced from the sources of the fruit and their corresponding microbiome, and how the organismal composition of the starters affects the baking process.
So, how do you answer these questions? After the whole sequencing process, how does the information get back to you?
Well, we used CHIME. It shows what bacteria and fungi are in the sample and how much of each type. For example, these are the bacterial results from fall 2020. You can see that most of the samples contained majority bacilli, but the fungal results were mostly unassigned. But Saccharomyces was present in almost all samples.
What is that?
That is the genus of fungi that includes many species of yeast, which makes sense. I want to focus on a specific bacterium, G. oxydans, which was in a peach starter from fall 2020, but not the corresponding control starter. G. oxydans has been associated with fermentation on multiple occasions. In regards to genetics, there have been some modifications to its genes.
Modify its genes.
Yeah, so a couple of genes have been found to cause metabolic deficiency. So they basically just replace them. There has been other research done, which can all be found on these sites, but I want to focus on my hypothesis. I thought that G. oxidans would be present in all fruit samples because, based off of my research, this bacteria thrives in a sugary environment and it is important for fermentation. So let's see if I was right. Here are the complete results from 3 samples containing peach, kiwi, and the control. But the next slide will approve or disapprove my hypothesis. The results are in, and I was wrong. Kind of. G. oxidans was in the peach sample, but not in the kiwi sample, as I thought it would be.
I guess kiwi is not as sugary as peach?
From what I have looked up, you're right. They are not a very sugary fruit. Now that this experiment is over, we're almost done. Now it's time to speculate. If I could get my hands on these samples, I would like to bring in another beverage or food known to go through a fermentation process to see if G. oxidans is present in it also. If it is, that must mean it is important to fermented foods, and it could potentially be sequenced to find exactly which genes are responsible for fermentation. After finding this out, maybe it can be genetically engineered with bacilli, which we saw was the highest presence in the starters, and we can amplify the fermentation process. If not G. oxidans, maybe another bacteria that I did not research.
Wow, that sounds intricate.
I know, and I could never do it. Just speculating. That wraps up this video about my project. Thank you to Shauna and anyone who watched and or listened, and I hope between my explanations, Shauna's questions, and the resources provided, all of the information was well understood.